Howdy, today I'll be presenting humoral immunity. The phases of humoral immune system start with the naive B cell recognizing an antigen through their B cell receptors, leading to the activation of B lymphocytes. Once the antigen binds to the receptor, the B cell is activated, inducing proliferation and differentiation of B cells to the two types of cells, which are plasma cells and memory B cells. Plasma cells are capable of antibody production and secretion, as well as neutralizing pathogens or marking them for other immune cells to destroy these pathogens. B cells that express IgG are capable of isotype switching, and B cells with high affinity Ig expression lead to affinity maturation. After the infection has been cleared, B cells become memory B cells that are able to have a faster and more effective response when re-encountering the same antigen. The stimulation of B lymphocytes by antigen can result in two main categories, including T-independent or T-dependent antigen mechanisms. B cells that are T-independent do not require helper T cells for activation, which happens when non-protein antigens such as lipids, carbohydrates, and nucleic acids bind to these B cells. B cells that are T-independent bind to bacteria and pathogens with repetitive antigen structures made of polysaccharides. B cells that are T-independent mainly have IgM and are plasma cells that do not live long. B cells that are T-dependent do require T cells for activations, which happens when proteins bind to these B cells. B cells recognize T-dependent antigens and internalize it for presentation through the MHC molecule to a helper T cell, which recognizes the MHC class II, activates and provides help to the B cell through the secretion of cytokines and receptor binding. Activated B cells that are T-dependent proliferate and differentiate into plasma cells that secrete antibodies. These antibodies are highly specific and they can undergo isotype switching and affinity maturation. Furthermore, these are plasma cells that live long and turn into memory B cells. The signals from innate immunity lead to the activation of the complement system by microbes. Once activated, it leads to the binding of a breakdown product, which is C3d, to the microbes. Afterward, the immunoglobulin receptor is able to recognize C3d and bind it to the CR2 receptor. The CR2 receptor is now able to deliver an activating signal to the B cell since it is attached to various proteins. Molecules from microbes may activate toll-like receptors of B cells when microbial antigens are simultaneously recognized by the antigen receptor. Heavy chain isotype switching is mainly for B cells that are T-dependent. Helper T cells stimulate B cells that express IgM and IgD to produce antibodies with different heavy chain isotypes. CD40L mediated signals and cytokines induce the activation of heavy chain isotype switching, and the cytokines produced by the follicular helper T cells are what determines which heavy chain isotype is needed for the specific antigen. IgM is mainly for complement activation. IgG subclasses provide neonatal immunity and are vital for opsonization and complement activation. IgE serves in mast cell degranulation and provides defense against parasites and allergic reactions. IgA provides mucosal immunity and therefore can be found in mucosal areas and body secretions, and they can neutralize pathogens at entry points. Affinity maturation occurs when the binding strength of antibody increases in response to a protein antigen with prolonged or repeated exposure to that antigen. Affinity maturation takes place in germinal centers of lymphoid follicles, and it is the outcome of somatic hypermutation of Ig genes in dividing B cells along with the selected B cells with high affinity to antigens. The regulation of humoral immune responses through antibody feedback is a crucial mechanism for maintaining immune homeostasis and preventing overactivation. Upon encountering antigens, IgG antibodies form complexes with these antigens. These antigen-antibody complexes can then bind to Fc receptors present on various immune cells, including B cells. One such receptor is FCY IgG4-RIIB, which contains an immunoreceptor tyrosine-based inhibition motif. When the Fc portion of the IgG antibody binds to the FcYR2b on B cells, it initiates inhibitory signals via the ITIM motif. These signals counteract the activating signals generated by the B cell receptors upon antigen recognition, thereby inhibiting or terminating B cell activation. The effector functions of antibodies include the neutralization of microbes and toxins, in which antibodies block the binding of microbes and toxins to host cells, the opsonization and phagocytosis of microbes, in which antibodies tag and promote ingestion of microbes. Antibody-dependent cellular cytotoxicity is when natural killer cells bind and kill antibody-coated cells. Complement activation leads to the lysis of microbes, inflammation, or phagocytosis of microbes opsonized with complement fragments. Vaccine development has led to many different types of vaccines, including subunit vaccines, conjugate vaccines, synthetic vaccines, viral vector vaccines, and DNA vaccines.